So we know when we talk about cell signaling that the first step is reception. So let's take a look at some examples of receptors in cell signaling. And so again, there are many, many different kinds. We're going to look at a couple major categories of receptors. And remember, this is just the first step, because that receptor, once active, is going to then pass on that information, and it's going to be transduced to ultimately result in some response from the cell. So most water-soluble signal molecules bind to specific receptors on the plasma membrane. Right? Remember, cells live in an aqueous environment. The cytoplasm is an aqueous environment. But that plasma membrane is our very robust chemical barrier. So we want to keep things outside the cell. How do we detect what's out there and get that information that it's out there into the cell? We're going to look at these receptors. There are three main types of membrane receptors that we're going to look at first, and these are G protein-coupled receptors, receptor tyrosine kinases, and then ion channel receptors. And if you're familiar with these three, it's enough to kind of get you started in the field if this is interesting to you, and you're going to come across this stuff again and again. So let's talk about G protein-coupled receptors. So it's a plasma membrane receptor that, surprise, surprise, works with a G protein. And it's kind of an on-off switch. So if a G protein is bound to GDP, it's off. When the G protein is bound to GTP, it'll be activated. All right. Now, the receptors, the G protein-coupled receptors, are characterized by having 7 membrane-spanning domains. So that's one indicator that you're in this family of receptors. So again, form and function and membrane topology. So there's a very specific loop of the protein that is found on the outside of the cell, and then another segment of that protein that is found on the inside of the cell. And when the binding site on the outside of the cell is activated by binding to its ligand, that's going to change the shape of the segment interacting with the G protein inside the cell. So these are real things. If you look at some of these actual signal transduction maps, you can see G-coupled protein receptors, different kinds, G-coupled receptor, G-coupled receptor. All right. So this is the real deal. Now, how does it work in a nutshell? So you have our G protein coupled receptor, and it's bound into the plasma membrane. It's an integral membrane protein. Now, the G protein is also bound to the membrane protein, but remember, because we're in the plasma membrane, that biological membrane, movement laterally is fine. So these things are floating around. Now, when the ligand, the signaling molecule, binds to my G protein coupled receptor, then it activates it. And that activity then increases the affinity of that receptor for the G protein. So when the G protein bumps into it, it binds to it and then activates and turns on the G protein, allowing it then to bind a GTP and kick off the GDP. Now, this is like a transient shot, and this is another way to kind of control a response, to gauge the intensity of the response. So let's say that signal molecule binds, it activates a G protein, that G protein binds GTP. Now it's floating in the membrane and it's active. So when it runs into the enzyme that its particular target, whatever target they may be on the pathway that it's signaling for, it activates it. But the GTP only binds momentarily. Ultimately, once we hit that cellular response, I'm going to now hydrolyze the GTP to GDP, and now we're back to the offset. So you can think about that, it was like just a little hit. It's like, hey, I honked my horn once. But now if there's an abundance of the signaling molecule in the exterior of the cell, that signaling molecule is going to be activating the G protein coupled receptor constantly. So that every time the G protein runs into it, it's on again, it's on again. And so now I'm honking my horn again and again and again and again. This is a big signal. Hey, pay attention. This is the real deal. It wasn't just an anomaly. All right. So I want you to think about that a little bit as a way to kind of control the amplitude of the signal. This is a mechanical way to do that. Right? So again, G protein coupled receptors are one category, one class of membrane receptors. Receptor tyrosine kinases are another. Now, receptor tyrosine kinases have receptors that are attached phosphates to tyrosines. Okay, their receptor tyrosine kinases trigger multiple signal transduction pathways at once because they're relatively big proteins. Now, the thing about these particular kinases is they only work when they're dimerized. And so by binding the ligand, what we see is we need to activate 2 adjacent tyrosine kinases that then have affinity for each other, form a dimer, which is a protein made of two components, and that dimer is then activated, and it's phosphorylated. So it phosphorylates itself. Okay, it self-phosphorylates, and then that phosphorylated version of the receptor can activate other proteins. Okay, and those proteins then relay the response into our next step of transduction. So again, just another family that you should be familiar with, just another mechanism. There are many, many of these. Now, the final type that we want to talk about is super important. And when you get into nerves and neurons and how they work, ion channels are going to be a big, big component of generating a nerve pulse, an action potential. But they can also, in this case, are going to act just as a simple gate. Right? So again, now I have my ligand on the outside of the cell will bind to my ligand-gated ion channel and open it. And that influx of a particular ion, whatever it may be, is the signal that can activate other proteins, okay, or other components of the transduction cascade. So literally, ligand causes the channel to open. Things, in this case, move down their concentration gradient. That itself is the signal. So oftentimes we look at this as a gate for sodium or calcium, and we can see these in the plasma membrane, certainly, but we're also going to see them internally in the ER. And when we get to second messengers, and when we talk about transduction, I want you to remember this idea of the ligand-gated ion channel. All right. Now, we talked about all of these receptors as membrane proteins, right? These are proteins that are embedded in the plasma membrane. They're feeling the exterior of the cell and waiting for a ligand to bind. But there are also intracellular receptors. So these are proteins that are free in the cytoplasm, waiting to bind a ligand, potentially from the outside of the cell. All that means is that that messenger, that information molecule, that ligand, has to be able to move through the plasma membrane, right? And so they're oftentimes small hydrophobic chemical messengers that then are going to diffuse through the plasma membrane and bind to the receptor, activating then a transduction cascade to result in a cellular response. So in this case, the receptor protein is free in the cytoplasm as opposed to being membrane bound. And an example like testosterone, which is a lipid, it can pass through the plasma membrane relatively easily, bind to its receptor, and then activate, in this case, alternative gene expression. Right? So remember, if something's going to move through the plasma membrane, it needs to be small and hydrophobic, okay, or uncharged, right, and small enough. But things that are charged or too large are not going to be a signal molecule for this intracellular receptor pathways.